We're jumping into Chapter 7, Neoplasia. This is an interesting chapter because Tax and Herman on your text, they do not go into great detail about specific cancers, so we are going to use a combination. I have added osmosis videos about some of the top common cancers that I think we see for grown-ups, and added that in addition to the text. But to start off with the neoplasia, they really want us to understand the physiology or pathophysiology of how cancer cells work, and therefore how cancers kind of come around to be. So they talk about the key idea that a cancer is a disorder of unregulated cell proliferation. Proliferation means to create new. It usually originates from one or a small number of DNA mutations due to environmental exposure or random chance. When you have an unregulated cell that can copy itself over and over, that reduces DNA protections, and that allows mutations to continue, and then they accumulate. In many cases, the mutations facilitate invasion across the basement membrane of the cell, and that allows distant metastasis. We're going to talk about the differences between proto-oncogenes and oncogenes, as well as tumor suppressor genes. This is an important key concept because you have to understand this idea that the proto-oncogenes are what we need in childhood, because this allows us to have rapid growth and development. If you think about the difference between an infant and a 5-year-old, that amount of growth in 5 years is insane. Like, you've more than doubled their size, all of that. And so we need those proto-oncogenes to drive that engine to quickly create cells to keep up with childhood growth. But then they should turn off. But if you have a mutation then that comes about that allows them to be reactivated, then they come back, and we call them oncogenes. And those allow repeated cycles of cell division, and then they don't follow the normal rules. Okay? So they don't have limitations like constrained space, lack of nutrients, lack of growth factors. They don't have the normal shutoff valve. And then we have tumor suppressor genes, which is codes for protein that stop the cell cycle, preventing cell division when a cell has abnormal DNA or pathology. So having tumor suppressor genes is a good thing. Having proto-oncogenes that get turned back on later in life to become oncogenes, not a good thing. This is actually always fascinating to me because the actual inherited predisposition to cancer is very rare. It's less than 5%. We don't typically get cancers just because of genetics. There's a large amount of cancers that come from not inherited genetics. Okay? When you have an inherited mutation that promotes cancer, that involves the tumor suppressor genes. And remember, if we talk about the DNA and then talk about the chromosome, and you have the two alleles, when one allele is mutated at birth, the cancer does not develop. But if you have that, so you have an abnormal allele and a normal one, when you take a second hit to that normal one, now something's going to happen, is you've lost your protection, and the cancer can now grow rapidly. So yes, you can have some genetics of mutations that can be passed down, but in terms of that flat out being the reason you got the cancer is very rare. You have to have the second hit. Gene mutations accumulate in the cancer cells due to genetic instability. The tumor cell genes are tracked during the course of treatment and used to guide chemotherapy. And I've put the link here for the osmosis videos about the cell cycle. The differences in the cell cycle is an important concept to understand, because when we get to pharmacology, you are going to hear about drugs that are specific to cell cycles. And so we have to have this good understanding of it. And maybe for some of us, this is a review. And maybe for some of us, you're like, yeah, I learned this once like 20 years ago. Okay? When we talk about the cell cycle, what we're really referring to is the steps that are involved in the cell division to replicate itself. The steps of the cell division encompass a complex system of these interacting molecules. Okay? So we have to have coordinated cell division in a controlled manner at a controlled rate. Okay? Your book gives the example that the rate of cell divisions can vary widely among mammalian cells. For example, that they give mature cardiac cells and neurons exhibit low rates of division, whereas cells lining the GI tract and blood cell precursors of the bone marrow will divide rapidly. So there's a lot of differences in cells based on their cell cycle, but they have to be regulated. So the cell cycle has distinct phases, and each phase has their signature pieces that are planned and should be carried out specifically in order. So our body has the ability to take cues in and change up and do processes to get the different phases of the cell cycle going. This image here in your book is figure 7.1, and it's showing us that during interphase the cell prepares for division. In G1, the cell grows, the organelles begin duplication, and RNA and protein synthesis are increased to complete replication of DNA in the S phase. So in B, this is the mitotic phase, the M phase, and this is the process by which the DNA is separated and it's going to form two nuclei. So let's review again interphase. So interphase is we go from G1 to S to G2. The cells spend a lot of their time here, either in interphase or in G0. In interphase, it's this period that they evaluate whether the conditions are appropriate to divide or not. This is also where they irreversibly commit to the process, and they will complete the necessary preparations for successful duplication. So you have three subphases: G1, S, which is synthesis, and G2. G1 is the most variable portion, and the amount of the time that the cells spend in this period is very individualized for what type of cell it is. Cells with high rates of division spend less time in G1 than cells with less frequent division. Why does this matter? If we talk about cancer cells, and we know that they are often rapidly proliferating, if I give a drug that targets rapidly proliferating cells, because I start to target G1, so I give them a drug to target G1 to stop rapidly dividing cells. That's a problem because now I could also affect my rapidly dividing cells of like the GI tract. Throughout G1, high RNA and protein synthesis rates support cell growth. The organelles such as the mitochondria and the lysosomes will begin their own process of biogenesis in preparation for providing each new cell with the necessary repertoire of organelles that will support cell function. And again, remember, it is during this phase that the cell makes a commitment to completing the remaining steps of cell division. The S phase is called the synthesis phase because it primarily involves DNA synthesis within the cells of within the nucleus. So during S phase, all 46 chromosomes are replicated, with each new copy remaining linked to the original by cohesive proteins. So these connected chromosomes are referred to as sister chromatids, which we heard about in the videos. Productions of histone proteins increase during S phase, and the DNA becomes tightly coiled around these proteins. And this creates a DNA histone complex known as chromatin, which helps organize, condense, and package the DNA in later stages of the cell cycle. Okay. When we are done with interphase, at the end of interphase, we're going to go into G2. And this is the spot where it's going to stop, and it's going to say, let's check for errors one more time. So it's literally like a factory moving down the line. And now we hit G2, and this is the inspectors that are sitting there and making sure the DNA is properly done. Following the completion of G2, the cell has reached a critical size. It has doubled its internal contents, replicated, and checked its DNA. Now it's ready to divide. So if you were to say, what happens at the end of interphase? It would be that now the cell has doubled in size and it is now ready to divide. So you have to have G2 complete before we can begin M phase, which is the mitosis phase. So there's the subphases of mitosis that are outlined for you in box 7.1, and these are very distinct phases as they're listed on there here, on your screen here. Really, that important point is that for prophase, this is when the chromatin condenses into chromosomes, and in each cell the microtubules are organized in the structure known as the centrosome. And your osmosis videos go through this quite well. In prometaphase, the nuclear membrane is breaking down and exposes the sister chromatids to the microtubules of the centrosome. In metaphase, the microtubules align the sister chromosomes towards the center of the cell, which is in this image here. So we're changing up the alignment. And the next phase of the mitosis cannot occur until all of these are properly aligned. Then in anaphase, we are shortening those outside microtubules begin to shorten, and they're literally now pulling apart the chromatids. So they're separated and pulled to opposite sides of the microtubule. So then into telophase, the two nuclear membranes begin to form around a now separated and condensed or decondensing chromosome. So at this stage of telophase, the replicated DNA has now been split and moved to opposite ends inside, but now the cell must still effectively divide the cytoplasm. And that occurs then during cytokinesis. At the end of cytokinesis, the cell cycle is now complete, and it now has produced two cells, each cell now with its own set of DNA and cytoplasm. So that's the life of the cell cycle. Now the cell cycle happens over and over and over again, and the really kind of cool thing is like you can merge in and out of traffic. So you can take an exit off the interstate, or you can come onto an on-ramp. So the cell cycles, they can come in and out of the interstate, so to speak, in the process of where they're at in that cycle. Okay. If a cell is leaving during the progression, so if the cell wants to leave somewhere in this process here or this process here, then that cell can't do that. In either state, the cell can still likely be functional and viable, but if you move out of the main interstate and you go to G0, that's the state where they're at rest. Okay, so this is not on this slide here, but an important part to talk about. It makes me think of like a roundabout where you could drive around in a circle, and within that, so you can just keep going round and round and round, and then within that roundness, you can merge on or you can merge off. So if they're in the active part of the cell cycle, they're driving around in the circle, at any point they can come off of the circle drive and they can enter into the state known as G0. When they go to G0, they go to that parking lot. To me, I would say the car is still running, but it's chilling in the parking lot. It's not going to achieve anything. Now, some of these cars on the interstate are only going to go around the circle once, complete their function, and then essentially be retired. So when they come off, they're going to go to the different parking lot called terminally differentiated, because they have matured and they have done their function and they're kind of done. They don't have to divide again. They may still be useful at their job, so they may be, let me rephrase that, during the cell cycle they're driving around the circle, and then they kind of go out and they do their job. But instead of ever proliferating and coming back into the circle, they just drive into the long-term parking, and there they sit. And they turn their engines off. So in that analogy, the roundabout of cars is the cell cycle. So when the cars are going round and round, they come in there to do the cell cycle and then divide. They can pull on and pull off pretty easily. Once they're divided and they need to leave again and now go perform their duties, they exit off onto the interstate and they drive around to the body. And then eventually they're going to come back and enter into the roundabout again for the cell cycle so they can divide again. However, some cars are going to come in and be made and leave. They're going to drive out into the country, they're going to do their job, and they're going to come back in on the highway. But instead of going into the roundabout, they're going to go over there and park in long-term parking, and they're going to shut their car off. That parking lot's called terminally differentiated. They've already carried out their function and they no longer need to divide. This is typical for cells such as cardiac myocytes and neurons, and the book gives a great example here. Once they've carried out their function, they're no longer going to divide, so it's difficult for cardiac myocytes and neurons to just quickly replicate themselves over and over. They don't tend to do that. So that's why if you have ischemic heart problems or ischemic blood vessels in your brain problems, and you damage the heart or brain cells, that often will result in long-term and permanent consequences. Also, some of the cars driving around the circle are going to be kinds of cells that have a limited amount of times that they can complete their division, because each time they do, they kind of get rid of one of their tail ends of their chromosome. So it becomes shorter with each division, and then eventually when you run short, you reach replicative senescence. So now these people, once they're done with their last divide, they're going to go park in another parking lot, and they're going to go in there, and they're going to turn their car off, and there they go. They're senescent, and they're thought to be an irreversible G0. They're like in the junkyard now. They're done. The book also talks about the liver cells to be different than the cardiac myocytes and neurons, because in that, those cells are programmed differently. They have to have a much higher capability to regrow, because our liver can regrow itself. So they can exit to G0, so they can be in the circle and they can pull off and idle in a parking lot for a while, and then at any time re-enter the cell cycle. So those guys are different because they can go out and rest and idle and come back in. Another example of cells that can act like that is our memory T cells, right? So during an exposure to the pathogen, the activated T cells will increase in numbers. So the T cells are going to go to the roundabout, they're going to enter the cell cycle, they're going to replicate and do their thing. But then when they're no longer needed because the infection has resolved, then a portion of these cells go, they exit the roundabout and they go to the parking lot where they just sit and idle quietly while they're taking a nap. Next we move on to control of the cell cycle. So we have a couple of different terminologies that we have to know here. The first one is we have to understand cyclins, cyclodependent kinases, and cyclodependent kinase inhibitors. So we have CDKs and CKIs. As you can see here, the cyclins determine cell cycle orderly progression. So cyclins promote the cell division. They're kind of a one and done. So after they complete the role, the cyclins are thrown in the trash. They're broken apart, degraded to prevent abnormal cell cycling. The CDKs are the workhorses of the cycle, and they are unable to function unless their level of their specific cyclin increases. And so the CDKs are what promote the cell division as well. The cyclin-dependent kinase inhibitors then, right, so we are inhibiting, we're stopping, those are activated by these signals, and they are going to halt CDK activity, and so therefore they stop the progression because they see a problem. Inhibit cell division. So we have two of these things that are going to help promote cell division, and then one is putting on the brakes and says, whoa, 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 we see a problem. In this image here, it helps explain that while cellular levels of the CDKs remain relatively constant throughout the life cycle, levels of the cyclins fluctuate with each phase. Cyclin D level rise through the G1, initiate increases in cyclin E, and you can see it's kind of a complicated thing, but as we're moving from G1 through the mitosis and back, these things are having their different activities. So it's important that they're able to power themselves to work through the cycle and continue to replicate, but it's also equally as important that we have things that are checkpoints or stoppers to do that. And we do call them checkpoints. So the checkpoints have to help stop things. It's kind of like throwing in the red flag in a football game to say, I want to go back to review the play on the field, right? So they will block the movement to the next cell cycle when they see that DNA is damaged. So they're like, whoa, 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 stop, let's go to instant replay. I have to make sure this DNA is appropriate. Sometimes this happens when the nutrients aren't sufficient, when energy is deficient, or the growth factors are lacking. So these listed here are some of those proteins, RB, P53, and P21. So we know that they operate at the G1 to S station. And they are considered tumor suppressor genes, and they're frequently mutated in cancers. Lacking these oversight proteins, the cell cycle progression can occur even in the presence of damaged DNA. So that's how we can perpetuate these abnormal cells. So the checkpoints are important. A couple of the different checkpoints that I wanted to add on here to talk about is what they do mention in the text. So one of them they call the restriction point, and that's where you're progressing through the G1 checkpoint. And that's going to ensure the presence of the necessary growth conditions and commits the cell to the remainder of the division process. So the restriction point checkpoint ensures that nutrients and growth factors are present and the extracellular environment is favorable. So once the cell has reached its necessary level of activation, it's able to move through that checkpoint and can proceed to the next part of the cell cycle. So that's a good example of what kind of checkpoint there are. So next they move into neoplasm properties. Okay, so this screen, believe it or not, can seem really simple, but it's really important. Understanding and using the correct terminology is a big deal. And I see this all the time, that we are using these words inappropriately. So a tumor is any abnormal mass of cells. It doesn't mean it's cancer. You can have a benign tumor, or you can have a malignant tumor. You can also have dysplasia, which is where their starch is showing some abnormality, but they're not yet a cancer or malignancy. Then also we can have carcinoma in situ. So it is a cancer, but it hasn't invaded. It hasn't leaked out of the basement membrane yet. And then we can have cancer cells of the blood and bone marrow. The malignant cells have certain characteristics that we talk about because it's important to understand what they look like and what makes them cancer cells, how we define them. Malignant tumors are named based on the tissue from where they develop. So the example here is a carcinoma is from epithelial origin. Of the carcinomas, we have squamous cell, adenocarcinoma, and small cell. You can also have a sarcoma. You can have hematopoietic leukemia lymphomas. You can have melanomas and teratomas. You can also have tumors of the brain that are like glial type tumors. So the terminology is very specific, and we have to use it appropriately. So some of the things that we see that are very characteristic, right? We have to know what identifies or how we identify a cancer cell, what the definition is, what are the common things they have in common together. One of the first things that cancer cells do is something with gene alteration. So remember we said we have our proto-oncogenes, which are really important to help rapid growth. They're highest during our embryonic period in childhood, and they essentially will just shut off when they're no longer needed. But if a mutation reactivates them, they become oncogenes. Oncogenes are bad. The conversion from proto-oncogene to the oncogene, oh my gosh, you guys, conversion from proto-oncogene to oncogene is what I'm trying to say, can result in this unchecked growth and proliferation. We can call these gain of function mutations, because when this gets messed up, we've gained function, we've gained activity, we've been released from the stop sign, so to speak, and we are allowed now just unregulated growth. Next, there are many protective genes that encode proteins that inhibit uncontrolled cell division. So if those are damaged, right, then we have a problem. And that is tumor suppressor genes. So we have loss of function. Remember, we have some kind of mutation now in both alleles causing loss of functional protein, and that can cause unregulated growth. This includes the cell cycle suppressors and many DNA repair genes. This is a visual example here of proto-oncogenes becoming oncogenes and the activity of tumor suppressor genes. So we have two essentially different types of genes that are at work. Remember we said we have these gain of function genes from proto-oncogene to the oncogene, and then we have the tumor suppressor genes. So when we have a proto-oncogene that becomes an oncogene, there can be a single mutation event that creates it. So this was normal. Now we've got that red spot here. It's changed. It's abnormal. This is activation of the mutation, and that's enabling the oncogene to promote cell transformation. So this is one way we do it. The other one here is that if we have a loss of function mutation. So now we have mutation that inactivates their tumor suppressor gene, right? And now we have two inactive mutant functional genes here, and that both of those can lead to cell division. One of the most commonly observed oncogenes is mutated RAS, and it's about 20% of cancers. So RAS, or RAS, is an important intracellular signaling protein that transmits signals that activate the cell growth for a brief time before being turned off. So if you have mutated RAS, that will result in continuous stimulation that will fuel tumor growth. Okay, so one of the proto-oncogenes that becomes an oncogene is mutated RAS. Another proto-oncogene code for the membrane epidermal growth factor receptor, which is EGFR. EGFR is a member of the tyrosine kinases. So EGFR is very common in cancers, and that causes unregulated cell division due to the continual activation of its signaling properties. So some of the drugs we're going to learn in pharmacology are tyrosine kinase inhibitors, TKIs, and that works by binding to EGFR and stopping this process. So we've talked about the uncontrolled proliferative signaling with the gain of functions. We've talked about those now. So next is evading growth suppressors. So the cells have many mechanisms that are going to regulate that cell cycle in response to activation of oncogenes or cell stress. The tumor suppressor genes controlling those processes include many cell cycle inhibitors and regulators. So when a tumor suppressor gene is inactivated, it's like it's getting turned off. And when you have those things turned off, you're at an increased risk of developing cancer. So the actions of a normally functioning tumor suppressor will inhibit proliferation. So that's a good thing. But when it loses its function, so loss of function, now you can lead to unregulated growth of cancer cells. Interestingly enough, over half of the human cancers will have mutations in the p53 tumor suppressor gene. So we now know that the p53 is vitally important in maintaining our integrity of the genome. When the p53 protein is not functional, DNA damage goes unrepaired and mutations accumulate in the cells, and now you are in trouble. The other one they talk about is the RB1. Genomic instability. This is referring to defects in DNA repair pathways. Because the DNA cannot be effectively repaired, the DNA mutations will accumulate. So now you have an unstable genome. You've got significantly increased risk of mutations. And those could include tumor suppressors, proto-oncogenes, or just pure cell death. So we have a couple of different characteristics of how the cancer cells act and how they proliferate. Next, we have their ability for enabling replicative immortality. I love that. Remember how we talked about that some of these, the DNA will have the telomerase at the end of the chromosome? And it's kind of like every time they replicate, they have to give part of their tail as payment. So what happens is they only have 10 pieces to their tail, for example. I'm just making an example. So after their 10th time of replicating, now they are done. They can't do it anymore. And so now they are going to go to the parking lot, turn off their engine, and just sit there, right? So if you have a cancer cell, it is going to avoid that cellular death, right? Because they can bypass this part. Some of them can actually increase the level of their coins. So the cancer will go, oh, here, you 10? Well, no, here's 100. Just keep going, right? So they bypass it, so to speak, and it could essentially become immortal. It's just going to happen over and over and over. So one of the drugs that we talk about later in the program is telomerase inhibitors, meaning they stop that process. Some of the cancer cells have the ability to resist cell death, means they just disregard apoptosis. So remember, our cells can be very much like an Apple iPhone. It's only going to work for so long before it glitches and dies. Cancer cell can resist this pre-programmed death. So they're allowing these damaged cells to continue to divide and divide and divide. So they can bypass that. So under normal things and conditions, apoptosis is a regulated and very programmed, very structural, or the word I'm looking for, very routine. It just happens. But again, when we now are going to do something because of a tumor cell, okay, they are going to be resistant of this mechanism. The next one is cancer cells have figured out how to promote their own blood vessel creation. So as the tumors grow, they require blood supply. And as they have this increased production of the vascular endothelial growth factor, they're going to continue to make new capillaries, and they just increase the blood flow to the tumors. Next, what can cancer cells do? The cancer cells can invade the tissues surrounding them and lead to metastases. So cells will revert in an epithelial mesenchymal transition, and you'll have adhesion to extracellular matrix will loosen, the matrix degrading enzymes are expressed, and the cells now become motile or mobile. Okay, so let's talk about this a little bit. So the cell, in order to metastasize, then it has to separate from the original tumor. It has to then invade the tissue, and then it has to enter and survive in circulation. So our blood vessels or our lymphatics, or even like in the peritoneal space. And then they have to make it to a distant organ where they have to adapt, survive, and proliferate. So in order to do this, the EMT is an important process that allows these transformed epithelial cells to invade tissues, resist apoptosis, and then spread. An increased expression of EMT transcriptional regulators results in a loss of adherence, meaning they no longer have the tumor or the cells being kind of staying where they belong, and they become mobile. The other thing a good old cancer cell can do is have evasion of our natural immune destruction. So the tumors will start to produce immune suppressive factors, including factors that inhibit lymphocyte proliferation and promote lymphocyte death. Isn't that lovely? Because remember, typically in our immune system, the cytotoxic T lymphocytes are capable of killing not only virus-infected self cells, but also cancerous self cells. So, having cancer promotes a state of increased generalized inflammation, and so with elevated cytokine levels both within a tumor and systemically, that may promote cancer cell proliferation. And lymphocytes are found within the tumor tissue, but they are not always bad lymphocytes. Regulatory T cells may be present in the tumors also. Cancer cells may develop characteristics that will activate the immune system inhibitory signals, and then that contributes to the downregulation of our immune responsiveness. So a long way of saying that around the idea of the tumors can now essentially turn off our own immune system. The cancer cells produce a ligand that activates the programmed cell death, PD-1, receptors on our T cells, B cells, and our natural killer cells, and that effectively stops our immune system from clearing them out. The PD-1 test is a test that is done a lot in cancer care. A lot of tumors are sent for genetic marking, right, so we can see what their genetic mutations are. And they look for PD-1, if it's PD-1 positive, because now we kind of know one of the ways that the tumor is working, and we can give immune drugs that are effective against PD-1. We have a lot of different, back to the right screen. There we go. We have a lot of different drugs now that are working with our immune system to turn it back on. So we have these CAR T drugs, which are great, and they're chimeric antigen receptor T cells, and they work to attack an individual patient's tumor, and they're going to find the checkpoint inhibitor that permits the lymphocytes to proliferate on stimulation. And now we have immune therapy, very much considered a hallmark of cancer treatment, because we know that when we can target some of these things, we can now be effectively killing the cancer cells. This screen here is a little busy, a little bit, but essentially what it's trying to tell you is that the environment in which the tumor exists or live in, there's a lot of different things going on that are going to impact how it grows, how it divides, and whether it can metastasize. And so when we can understand the tumor microenvironment, we can continue to work on finding drugs that can help stop these structures, okay? So this one here is showing kind of the ways that the tumors can progress. So the tumor is made up of a bunch of different cells, right? We have the cancer cells, we have the stroma of the original tissue, we have lymphocytes, macrophages, dendritic cells, and some of the cells are in that phase of EMT trying to become mobile. The tumor region that is closest to the blood vessels up here, they have a normal oxygen supply. The tumor region at distance from the blood vessel has hypoxic and acidotic cells that cause cell stress. That helps promote the acquisition of cancer cell immune resistance and makes the immune tolerance parts of the cancer cell. So what this is saying is that we have the cytokines, chemokines, metabolites at play. We have the stress tumor conditions because it's hypoxic and acidotic, which the tumor loves. The tumor cell adaptations can be occurring that are going to help mobilize and cause invasion and tissues, or invasion and metastasis, excuse me. So having our typical cells is typical physiology. It becomes pathophysiology when the cells mutate and now become cancer cells. So the next part of the book goes into talking about this pathophysiological alteration at the systemic level or at the local level that we will see when people have cancer. Chief among them is going to be inflammation. So you're going to have elevated cytokine production that may cause fevers and suppress the appetite while also promoting clotting. Hypercoagulability is also common in cancer, particularly in early stages. Some people will actually be diagnosed with their cancer because they present with a DVT or a PE. Fatigue is often also common because of the inflammation, and it's often disproportional to what it could be. Like, it doesn't make sense why someone is so fatigued. That can be very common. Poor appetite and wasting can occur in advanced cancer. Endocrine-related syndrome can result from tumors that are ectopic sources of hormones or hormone-like substance, right? So what we're saying is that clinically, there's going to be a bunch of different ways that we can detect cancer, or we are going to have different clinical clues that we should pay attention to that could be signaling cancer. So if someone has fever, malaise, anorexia, we have to consider cancer as a differential. If they come in and they have a blood clot for no reason, if they have disproportional fatigue, if they just can't eat, they have no appetite. Or sometimes people have other weird things like SIADH or even just plain old hyponatremia because of a cancer somewhere that is causing these endocrine changes. The book then jumps into tumor lysis syndrome. Is a clinical thing that we may see, but it's actually usually caused by cancer treatments. So when we initiate chemotherapy, we target a bunch of cells throughout the whole body, and as you know, we can kill them quickly sometimes if it's a potent chemotherapy, but when those cells all die at once, they essentially break down and degrade, and they cause like trash overload, so to speak, and that overloads the body with these damaged cells, and this can cause fluid and electrolyte imbalance. Although it's considered spontaneous, like there's no guarantee that people are or are not going to get it, we are pretty good at preparing for it because it does happen in certain types of treatments, most likely, so we can kind of pre-treat it. When you think about tumor lysis syndrome, you have to think about altered electrolytes, too high or too low. When those things happen, you can get like excitable tissue, so we can think of things like cardiac problems, cardiac arrhythmias, or seizures. Also, I always think of it as the tumor lysis syndrome just plugs up the kidneys and can precipitate an acute renal failure. So, in order to head this off at the pass, for certain chemotherapies, they're going to assess your risk for it, and then they will treat it preemptively with hydration. It's typically the main thing. They're just going to keep those kidneys flushed, and then they carefully monitor you, and they're monitoring your labs. The book then jumps into biological aspects of gene mutations and cancer risk factors. We did say that there are cancers that can result from our inherited things, right? We call those germline mutations because they occur in the DNA of our germ cells, such as our eggs and sperm. Inheritance of the germline mutation results in every cell in your body is going to have that mutation. Okay? It's a minority of cancers. If you do have a germline gene mutation, it's often in the tumor suppressor genes and DNA repair genes. The other type of mutation that we can see is somatic mutations, and that's spontaneously or as a result of certain exposures. So this is skin cancer because you were repeatedly exposed to UV rays. Then we would call that a somatic mutation. But here they list often found exposures or things that we know do help promote cancer. Smoking, the UV light, older age, our diet, alcohol intake, obesity, and then if we're exposed to other chemicals like asbestos. Smoking is the leading cause of lung cancer. That's no surprise, right? Some of the specific hereditary cancer syndromes that we have to know about is Lynch syndrome. And this is typically because of a defective mismatch repair gene, and this causes the non-polyposis colorectal cancer. Then we have the Li-Fraumeni syndrome, which again inheres that p53 gene mutation. And then we know about BRCA1 and BRCA2. Those function like tumor suppressor or DNA repair proteins. Think of BRCA1, BRCA2 not only with breast, but also ovarian and fallopian tubes, and prostate. So men with positive BRCA1 or BRCA2 have been associated with an increased risk of male breast cancer and prostate cancer. B-cell lymphomas can arise from a chromosomal translocation activating the BCL2 oncogene. Chronic myeloid leukemia, this is something that I remember having on board exams, is caused by a chromosomal translocation between 9 and 22, which is the Philadelphia chromosome. Then you get the abnormal BCR-ABL gene, and that results in the protein tyrosine kinase initiation and causes unregulated cell division. And your book or your videos talk about that as well. Then we got the good old cancer that's caused by viruses. So these outline well. These are the things that you would want to know for like a board exam as well. Epstein-Barr can cause Burkitt's lymphoma. Hepatitis B and hepatitis C can cause hepatocellular carcinoma. HPV, certain strains, will put you at risk of head and neck cancers and anogenital cancers. Okay? Which is interesting because hepatitis B and HPV can be prevented by vaccines. So we want people to have those vaccines because if they don't ever get hepatitis B or they don't ever get HPV, we are reducing their chances of cancers. There are many, many types of HPV, but we are worried about two specific subtypes of 16 and 18 because those are the ones that are most shown to cause the head and neck or genital cancers. The viruses of the HPV can make E6 proteins, and those interact with the P53 protein and cause it to degrade. So now we've kind of turned off the P53, and this is going to lead to uncontrolled cell proliferation and ultimately creation of cancer. We jump next into the idea of genotyping in cancer for diagnosis and treatment. It's actually kind of cool because now cancers can be genotyped. So you'll see like the tumor or the biopsy was sent for genotyping. And we have a wide range of techniques that we can do that. Okay? For years, we only had surgery, radiation, or chemo. And the chemo was kind of a broad sweep. Non-specific chemos that just killed a bunch of cells. And that wasn't the best. We had these limited options. The treatment made people just as sick as the cancers. But now, instead of giving non-specific chemotherapy, we can genotype the cancer. And if we can find these mutations and use targeted drugs for them, typically the drugs are better tolerated and they're more effective with less side effects typically. The example that they're talking about here is that if we can identify the BCR-ABL fusion gene in CML, that led to us creating imatinib that we knew could be an effective treatment. So once we had imatinib, well then we started using it on other types of cancers and found that it also was effective. The other one we think about is the HER2 gene in breast cancer. It's in some other cancers as well, but we know that oftentimes we describe breast cancer as HER2 positive or HER2 negative because it is such a big deal. If they're HER2 positive, the treatment becomes very different, and we use these drugs that are listed here. Very cool. This is a pretty good condensed version of pediatric considerations. The most common childhood cancers typically tend to be leukemias and lymphoma. Adult solid organ cancers often originate in epithelial tissue and are the carcinomas. That is what can separate them from pediatric cancers. Very rarely does pediatric cancer come because of an environmental exposure. Pediatric cancer is also more common in the nervous tissue and the connective tissue. There's some other unique ones like Wilms tumor or retinoblastoma. And those are pretty straightforward to read in your book. Box 7.2 presents signs and symptoms of cancer in children. That's a little bit different than what we look for in adults. And they give a couple of examples of these different conditions. Then we have, those are the signs and symptoms of the cancer in children. When we talk about gerontological considerations, again, now we have to think about the environment. We've been around a longer time when we're getting older, right? So the longer time of exposure now has probably led to a lot more things that we have been exposed to that are bad. And as we accumulate those, we think is what ultimately leads to the mutation. We tend to see a decline in our own ability for DNA repair mechanisms as part of aging. This one here is a good illustration of what we're trying to say. There are multiple sporadic DNA damaging exposures, so we're thinking free radicals, carcinogens, or the viruses, and these will lead to altered expression of our oncogenes, tumor suppressor genes, and our apoptosis genes. Okay. So here, if you think about it, so you can see there we've had some free radicals, some carcinogens, some viruses. We've had an increase in our oncogene activation and mutation. We've lost the ability of our tumor suppressor gene to work, and we've lost our apoptosis gene. So initiation, you can see there with the arrow, that's of the altered phenotype with increased cell proliferation. Then we have the accumulation of additional gene alterations, which ultimately leads to promotion of the transition to malignancy with gradual acquisition of invasive and metastatic capacity, and the tumor will continue to expand. All the while, these things are occurring against the natural backdrop of declining host resistance. When factors favoring malignancy outweigh the factors inhibiting malignancy, well then, cancer develops. So the reasons why people that are aging have more cancer is because we have a favorable environment for more cancer. So we think that there's a longer time of exposure to the environment. We have that natural decline in DNA repair mechanisms as we age. Inevitably, we think we begin to damage our own telomere shortening, and therefore we cause our own destabilization of the DNA. Our immune system is not as good, so it's not surveilling as much. And we have an increase in senescent cell number and progression to the senescent-associated secretory phenotype that then promotes chronic inflammation. The book talks about immunosenescence because the immune system is our major defense mechanism against developing cancer. And remember, the immune system also functions as monitoring for tissue homeostasis so that we are ready to protect against invading pathogens. Also so that we can find damaged cells and take care of it. So the immune system works by eliminating or suppressing the viral infections to protect us from virus-induced tumors. But again, as we age, we have a decline in that. The immune system will help eliminate pathogens and promptly resolve inflammation to prevent an environment that is conducive to the development of growth factors. So as we age and our immune system begins to decline, we lose the ability to resolve inflammation. Therefore, we lose that protection of that. Our immune system typically will identify and eliminate tumor cells by the recognition of specific antigens. But as our natural decline in our immune function with age, we lose the ability to eliminate tumor cells by this. There's also a theory that the gradual decline in the thymic output of T cells may be another aspect of the aging process that can aid the development of cancers through immunosenescence. Our decline in our functional immunity is not only more permissive of tumor formation, but also promotes it because we have a chronic low-level inflammation when our immune system can't work to turn it off. We have age-related decline in T cell numbers and age-related decline in responsiveness of our immune cells. We talk more about that we talked about that in Chapter 6. this one here. The other important thing that happens with aging too is we have changes to our stem cells. So our body should have an ongoing proliferation of stem cells. Our stem cells should also be able to differentiate and also self-renew. So they are the major player in our homeostasis. Our stem cells and their ability to produce what we need is what we need in order to stay stable. Well, natural aging brings a cascade of changes in this homeostasis, one of which is a decline in our organ functions that affect the hemopoietic system, primarily our stem cells. Our stem cells in our bone marrow work all the time. They're constantly replacing blood cells that are constantly being lost, sometimes due to splenic destruction, sometimes just from tissue utilization. So our stem cells have to continuously regenerate to keep our blood and our immune system up and running. We tend to have our own hematopoietic reserves, especially in our spleen where the things can be stored. But as we age, we deplete our natural reserves, and we have lost our ability to renew as well as we did before. So now we don't have as many in storage, and we struggle to make new ones as we age. So now we're in trouble. The ability of our stem cells to differentiate into the different cells that we need, right? Because we have precursor cells and we need to produce those, and then they are triggered to divide into the certain types of cells after that, like downriver. But upriver, then, what we're talking about is that differentiation into those different cells isn't working well. And as we age, we tend to produce the myeloid cells better than the lymphoid cell production. Just because the stem cells are not functioning well doesn't always mean that we lose the numbers of them, because they have a long lifespan. However, because they have a long lifespan, they are subject to oftentimes having damage from both intracellular and extracellular sources. So think about oxidative chemical reactions within the cell that happen with time. Our stem cells undergo repetitive over and over and over DNA replication. And sometimes when you have such repetitive motions, you get random errors. So as a result, genetic damage occurs and actually can accumulate. There's an instability of the stem cells to exist in great degree in bone marrow of the older adults. And that suggests a progressive decrease in the repair process, and this all together leads to genomic instability. Damage to DNA occurs in the aging process. It is a part of aging from prolonged exposure to our internal and external factors. We know that our DNA repair mechanisms start to falter and don't work as well with age. So if our DNA cannot be repaired, our mutations will accumulate and they'll continue to grow. And that leads to the increasing incidence of transformed and malignant cells. This can lead to a condition called myelodysplastic syndrome. I often would say that myelodysplastic syndrome, to me, could best be described as a failure of our bone marrow, mainly from age. Doesn't always mean that it's a cancer. It just means that we have a decrease or defect in the process of our bone marrow to keep up with proliferation. So, because we know that things are going to change and our risk factors for cancer will increase with age, this impacts how we do our screening and our surveillance. Oftentimes, when we think that putting somebody through that test or that screening process, the benefit has to outweigh the risk. Well, sometimes if you have frail older adults, now all of a sudden it's not worth the risk to them to try to screen for cancers that may be even less likely to happen in that decade of life. So we have to take into consideration the functional status of the patient, what their performance status is, or are they frail. So we see this in things after the age of 75. We really slow down on colonoscopies and cervical cancer screening. The other hard part about cancer, and when we get to be older, that will impact how we practice, is the simple fact that elderly people do not tend to tolerate treatments as well as younger people. They're more prone to side effects and they can suffer more greatly, and they don't always have a lot of reserve. So if they get 3 or 4 days of nausea and vomiting, they're not going to handle that as well as a 40-year-old getting 3 days of nausea and vomiting. The good news is that as we have created some more of these novel therapies and the immune therapies, like I said before, we tend to tolerate that better. So now that's nice because maybe we have more options for the older adult than just the non-cell specific chemotherapies. And then in your book you have your bridge to clinical practice, which I think is wonderful. This is again where we're beginning to foreshadow what you're going to be learning as you move forward throughout the program, where we start to look at the history and the physical, their assessment, their diagnostic studies, their labs, and then start to talk about some of the things that we could do for their plan of care. So again, to review some of the key concepts we've talked about, the cell cycle is a series of events in the life of a cell which promotes the cell to be able to replicate and divide. We have those different stages that we talked about, G0, G1, S, all those G2, and then goes into mitosis. Remember that the cells can spend varying amounts of times in each. They can go into G0 sometimes and lay dormant. We have to know that the certain cells have different signaling, and that the different levels of the cyclin proteins will change the cell signals and affect how they're able to enter into the cell cycle. After a number of divisions, cells may enter replicative senescence, after which they will not re-enter the cell cycle, and they go to that other parking lot to die. Control of the cell cycle is dependent on needs, but also on signals internal to the cell. We have cell cycle checkpoints that are there to assure cell readiness and healthy status, making sure that everything looks good in the DNA before it progresses to replicate itself. The protein P53 is a critical signal for blocking cell cycle progression when DNA damage has occurred. Cancer is a state in which the cell proliferation proceeds uninhibited by our usual control mechanisms, and we create tumors or a large amount of progressively abnormal cells. We name malignant tumors by their tissue of origin, and metastasis is the ability of the tumor cells to break off from their original site, travel through the blood or lymph to a distant organ. Proto-oncogenes may be activated into oncogenes, which is a gain-of-function mutation, and this is going to stimulate unregulated cell proliferation. The tumor suppressor genes may be turned off, so now we have loss-of-function mutation, and that reduces our protective reactions such as DNA repair when the damage has occurred. We talked about the different hallmarks of the cancer cells, right? So what are they? Often cancer cells will have 1. Uncontrolled proliferative signaling. 2. Evasion of growth suppressors. 3. Genomic instability. 4. Telomerase activity. 5. Apoptosis evasion. 6. Angiogenesis promotion. 7. EMT, that contributes to invasion and metastasis. 8. Evasion of immune destruction. And 9, ultimately metabolic alterations that will promote cell survival. We have cancers that can be in an organ, but cancers can also be in our bone marrow blood cells. think leukemia, lymphomas. Cancers can be localized or generalized. Cancers tend to produce systemic changes that lead to certain signs and symptoms, including profound inflammation, hypercoagulability, changes in our blood count, mucosal lesions, fatigue, fever, pain, anorexia, even changes in mood and changes in sleep. 90 to 95% of cancers are going to result from those sporadic DNA mutations. Lots of them are proto-oncogenes that turn to oncogenes. Hereditary cancers are going to be found in a germline mutation. We can also have cancer that can be caused by virus exposure. Our advancement in science has led to genotyping of the tumor cells at the various stages of their disease, and so we can hopefully find a targeted therapy that would work for that cancer. When we take into account pediatric and dermatological consideration, for pediatrics, the tumors are often in the blood and bone marrow, much more commonly. They have their own different kind of cancer screening questionnaire to think of. With adults, we tend to think of solid tumors, and we tend to think those are caused by repeated exposures to the environment. When we talk about aging in cancer, we know that cancer increases with age, and that age then also can impact how the patients can be treated for their cancer. There's possible explanations for age as the risk factor because we think there's this increased number of years that we've been exposed to the environment. Inflammaging, that chronic inflammation, increased numbers of sporadic DNA mutations, and our immunosenescence happens. Our immune system starts to falter and can't work as well. Thanks for hanging in there with me. That's our review of Chapter 7.